having me, Daria, in this restoration session, and I'm excited to be here, yet feel completely unqualified to be here, just to be candid with you. I was trained as a molecular systematist and then as an evolutionary biologist focusing on plants, and I blame my students completely for what I've done. Very few of them can make it because they're in classes today. Their classes are getting in the way of their education, so I'll blame them because I had the opportunity and was approached by Marianne Showers at the California Fish and Game at the time to try to help understand some questions in Erysimum and the wallflowers. As a new faculty at Santa Clara, I thanked her for that and dove into a project for a couple of years on the pollination, mating system, some population genetics of a couple rare species in the Bay Area. In doing so, the students were very helpful throughout the process, asking lots of questions. In fact, it wouldn't have happened without them. And finally, one of my Santa Clara students at the end said, the paper was published, the reports were submitted, I had given a couple talks, and the students had done really well, and they said, so now when do we save the plants? And I thought about it, and I said, well, somebody else is going to do that, so let's move on. And I waited, and then realized that nobody else was going to do anything about it, so the students are to blame for our attempted reintroduction here of Ben Lomand Wallflower. But it not being a trained restoration biologist, I had no place to start, so I thought I would start with some kind of philosophy. So here's a philosophical moment, and we all speak Latin, I understand, so I thought this was like a community building moment. Primum non nocere. That was Hippocrates' approach to what you might know as the Hippocratic Oath, or first, do no harm. That was the most important thing for me going into this project, is just to not screw things up out there in the sandhills. I've been doing a lot of carpentry work recently. My mother-in-law is moving to the backyard, so maybe a more practical and less like ivory tower approach, using Latin. Measure twice, cut once. It's a really important approach to not only carpentry but also restoration biology, I feel. So you'll see a lot of measuring going on ahead of time and then a little bit of preliminary data. This is where I'd like to take you. I'd like to motivate us with some information on wallflowers in general, because they are kind of my passion. And the Erysimum capitatum alliance in particular here in the West. Then talk a little bit about preparing for the reintroduction, and then if that's not enough, let's plan for the reintroduction. And finally, we actually did some stuff, and I'm going to tell you a little bit about what we found so far. So, Erysimum's pretty famous. The 5th largest genus in the Brassicaceae, which is one of the top 5 families in California. Around the world, you only find it in the northern hemisphere natively at somewhere between 200 and 250 species. It's definitely monophyletic. In fact, it's in its own tribe, and nobody knows where that tribe belongs, but we're now pretty sure it's sister to Arabidopsis, which comes in handy at times. It's really well known for being in exposed habitats, and edaphic endemics proliferate in the genus, but in doing so, you're exposed to a lot of herbivory, so not surprisingly, they're pretty well defended. They've got these oppressed metaphyx trichomes and some mustard oils in there, glucosinolates, as well as some cardiac glycosides. Oppressed metaphyx trichomes was confusing to me, and I knew I had a captive audience here. So Amelia Bethke, one of my students, did some nice drawings, and there's a picture of them. They are oppressed, their hairs. They are oppressed, that is, they're laid down on the tissue, typically on the stem and leaves. And they're metaphyx, that is, they're branched and branched in the middle. That's unique to all wallflowers. But the cardinali diversity is also pretty fascinating. These have heart medication capabilities. They are probably not there for the heart, but instead to prevent beetles like this from nibbling on them. Some of the diversity shown here from an old TLC from 1982. Some work by Rodman that was fascinating, how much diversity there is in the cardinalides, and some really interesting questions about plant defense that I can't touch on today. But thankfully, Tobias Zeus has done some great work that should be coming out shortly on that. We kind of wanted to know a little bit about Erysimum, so we started with taking a lot of field trips and collecting a lot of tissue, and then we wanted to see how they were related. We went to all 3 genomes here, and we came up with an estimate of relationships with some outgroups here in the Brassicaceae. You recognize Arabidopsis here. Erysimum is definitely monophyletic, and the red branches are strongly supported. That's good news. We were able to use some data from Mark Bielstein's work, applying this fossil. That's a Thalaspi primiveum, which is about 30 million years old, and belongs somewhere right here around the base of Thalaspi arvense and alley area, to help get an estimate of the date. And that puts the whole genus at about 2.7 million years, which was pretty exciting when we bumped into that, because if you calculate the diversification rate of that, that puts the crown or stem, depending on which one you want to go with, way out beyond the average diversification rates that you find in things that you should recognize here, like mammals and insects and reptiles. A couple of basidiomycetes have some estimates that are pretty far out there, but we can claim to have diversification rates twice as high as the Hawaiian silver swords and approaching the like Tanganyika cichlids, and very similar to those lupins that we heard from Teresa this morning. So a pretty exciting and hopefully a motivating group. In fact, maybe we want to think about preserving adaptive radiations in addition to old lineages. Phylogenetic diversity measures focus primarily on the length of the branch. The oldest things are prioritized, but maybe these are the motors, the engines of diversity. But that's another topic. Here's Tobias Zeus's work across wallflowers in the world, and he's measured their transcriptomes. That's all the genes expressed in the leaves. It's mind-boggling, and it should be coming out shortly. All I want to point to you is he only had a couple of North American taxa. They do come out together, and there's pretty strong biogeographic patterns, as you can see from the coloration there. So let's focus in on that North American clade. Here they are. They were named the Erysimum capitatum alliance by Robert Price in his PhD dissertation, which was unfortunately never published in 1976. So I've been leveraging a lot of his thesis work for this. There's about 25 taxa. There's a lot of edaphic endemism. You should recognize these blue-green soils, of course, and some sand hills and some sandy beaches over there. And just for fun, let's see how many wallflower populations or species we can bump into within a day or 2 days drive. So if we start here around Santa Clara University, which I think takes us right around here, we can drive up to the top of Mount Hamilton and see Erysimum capitatum, an unusual one, and the shales there. You can go down to Coyote Ridge and see Franciscanum. Maybe hop down to the Monterey Peninsula and see some Menziesii, a little bit of Ammophilum and more Menziesii, then some weird Menziesii called Yandonii, which is suspiciously succulent, maybe some more Ammophilum, and maybe stop at the sandhills on the way home and see Erysimum teretifolium, which is what we're here for. And if you aren't happy with that, maybe we drive up north on the peninsula and stop and see some more Franciscanum, which has an unusual shape to it on Fulgus Ridge, and then there's some capitatum at Devil's Slide, and then another Franciscanum with a unique name that probably isn't Franciscanum, and then maybe come back through Santa Cruz, see a couple more populations. Anyway, there's a huge amount of diversity, and it's right in my backyard, so it behooved me to get involved. Some more student drawings here showing you some of the fascinating leaf shapes, and I want to point out teretifolium because it's terete leaf shape here. We tried our best at trying to reconstruct the phylogenetic relationships of the alliance. It's definitely monophyletic, that is, there's only one shift from Eurasia to North America, but within that it's a big nightmare, and I won't go through the details except to point out that teretifolium samples from across the small species range in the sandhills is pretty monophyletic. We tried to sequence the whole chloroplast, and we did, unfortunately. There's two strong clades. There's a teretifolium clade here, and then there's another teretifolium clade down here, and I don't know why, so let's just keep moving on. Luckily, we don't have a lot of these gray areas. There's strong morphological differences. There's a lot of allopatry, and the edaphic endemism helps as well. So let me tell you a little bit about teretifolium here. We're going to go into some microsatellites, some mating system study, and some pollinators. The microsatellites we borrowed from some Eurasian species, but that works pretty well. And we looked at the physical distance between populations measured in meters. We're in Santa Cruz County here in the Sandhills, and the FST, which is the estimate of population differentiation. And you'll see if you just look at the dark line here, it's not significant. There's a little trend going on. But what happened is these points are out of place. And we were talking to a naturalist who was really trying her hardest to do her best, and she was going over to these populations and taking seeds out to try to supplement these populations, which was super sweet of her. Unfortunately, there was some questions about local adaptation that we were trying to solve, and that may have complicated that. When you remove those, you get a really strong isolation by distance. And we refer to this in the paper as unsanctioned restoration efforts, but I think it's more of Johnny Appleseed, and you should recognize that I say that with the utmost respect, that Johnny Appleseed is a national hero. Most people would consider him a conservation icon. But be aware that, if you go appleseeding, some people can detect where you've been. All right. With that, let me tell you a little bit about Miranda Mellon and her mating system study in collaboration with San Jose State. She got a master's degree through some funds that we got for Section 6 from the Department of Fish and Game at the time with Marion Showers, and then thankfully Sherilyn Burton came in and helped us finish it off. We grew wallflowers from 4 of the largest populations. There's only about 7 left, and 4 of those, the Bonny Doon one being geographically distinct was the big question. And we looked at self-pollinations, within population pollinations, and between population pollinations, and we looked at the number of seeds that they produced, wondering if, like a mini mustard, this is self-incompatible. And they made very few seeds in the greenhouse when we selfed them, but they made some like 7 to 8 times more seeds, so they are strongly self-incompatible. And that led us to the microscope, where we would observe the pollen grains at the top on a self, not growing down to the pistil, through the style to the ovules, and then an outcross, a really strong signal of growth there. That's consistent across a bunch of different examples. So this is a problem when you are rare, and you get even rarer because of mining of the sandhill soil, and now all of a sudden you can't mate with yourself. You better have some pollinators around, so we went out and recorded pollinators with these cameras, focused here on Erysimum teretifolium over 2 years at 3 populations, and I don't know if our media services will ever let us back, given all the sand that returned with those cameras. But the popcorn was generously provided by the local movie theater, and the students watched some 1,108 hours of movie footage. It was fascinating stuff, 506 pollinator events, about a half of a visit per flower per hour. Here's some of our greatest hits. And we had a diversity of visits from things including, small bees and flies and big bumblebees and butterflies and moths. Those are the big days, but often there was just an aphid sitting there, and you had to watch it, and watch it, and watch it. Oh, touch the anthers, but watch it, touch the stigma. Okay, it counts. We have an aphid, and I think we also have a spider in there, yeah. All right, I got to point out Jody McGraw, who really made this restoration possible, and she is a real restoration biologist, and she did her PhD at UC Berkeley, and she studied the more ecological concepts underlying Erysimum teretifolium. Her point is that gopher mounds are one way that teretifolium stays in the sandhills. It turns out teretifolium needs disturbance, and her great contribution, I think, amongst many other things, being a spokesperson and single-handedly almost responsible for sandhill preservation, is that disturbance actually increases native percent cover, unlike a lot of the lore at the time. So let me tell you a little bit about the planting stages. The Bonny Doon Ecological Reserve is in Bonny Doon, just outside of Santa Cruz, and here are some of the charismatic microfauna that you could find only in the sandhills, Zante sandhills of Santa Cruz Mountains. This is the bandwing grasshopper. Here is the June beetle, and here's some charismatic macrofauna. This is the Santa Cruz cypress, and I think there's some manzanita people in the audience, so I would be remiss without mentioning the silverleaf manzanita. Some charismatic microfauna. This is the Coryxanthe pungens, a rare endangered species, the other listed state and federal species from that area. But we're here to look at Erysimum teretifolium. This is a wonderful place. I have to pinch myself out there when I'm working. We designed an experiment, thanks to Jody's brilliance, where we would put seeds in the center. Those would be all from Bonny Doon, because we wanted to make sure we didn't contaminate it any more than it may have already been genetically. And then we would put juveniles around the outside who were transplanted, which would come from 4 different genetic treatments. That would be within population, that Bonny Doons, or maybe Bonny Doon by something else, or something else by something else, and then all pure something else. I say that because there's 3 other populations, and they all behave similarly statistically. We raked and tilled the plots. Some of them got raked, some of them got tilled, and they were all enclosed to keep as many herbivores out as possible. We did it over 2 years, and from this picture you can see not only our generous volunteers, but you can also identify the ecological treatments by their weapons. We have the shovels for the tilling and the rakes for the raking. In fact, in the sandhill chaparral, we required additional weaponry. That's Terrace Castine, who is helps support the Section 6 Reintroduction grant under Fish and Wildlife that we're California Fish and Wildlife that we're currently using, and Alyssa Wilson, who's my techie, is doing a lot of the work. Oh, and this is Alex Rickard, who's here today. He's a master's student at San Jose State, and he was a generous volunteer on almost all events, but he also has a very fascinating poster on bird nests as botanical time capsules. Please make sure you see that tomorrow morning. Enough plugging of my students and helpers. We tilled these down to 1 foot deep, and we made plots and excluded them, and here's some quick pictures of what it looked like. The first survey was going to be under snow. This is a little challenging to get there in the first place, and now that we're there, it's a little challenging to actually measure the plants. We found that the calipers quickly failed at temperatures below 32 degrees Fahrenheit, and the plants were even hard to find. But once the snow melted, which was relatively quickly, we were able to make some measurements, and here's what we got. We got really high survival. Watch the y-axis here. It's almost 100% survival, whether you were raked or tilled, but there's a little difference in the tilling versus the raking, but it's really subtle, even though those graphs suggest otherwise. We also see a much larger proportion of reproduction in the tilled, and that is about 3 times as high, but very few plants went to reproduction. And the treatment, we measured the longest leaf length and how much it changed from when we planted it versus not. It's a little bit higher than that tilled, but the take-home lesson is the plants really like disturbance, and if you dig a hole 1 foot deep and then put the plant in it, it really likes that. We're not sure why. But if you just rake the top, they don't like it as much. We also looked at the populations. There's the Bonny Doon population doing really well, and some of these non-Bonny Doon populations not doing so well, but keep in mind almost 95 to 100% of plants survive at each plot. Similarly with reproduction, most of the Bonny Doon, more Bonny Doon plants reproduced than others. The longest leaf length was not significantly different. Last thing was that we had to cull plants. This is really painful to share with you, but here's a picture of it. So the plant wasn't culled, but its reproductive attempts were. That is, we didn't want any seeds to escape, and although it's Self Incompatible, we didn't have the time to bag every one, so we had to cut the plants that were not from Bonny Doon this first year until we can prove for sure that there's no significant increased fitness of those guys. The seed plots are right here, and we don't have those well analyzed, so I'm going to skip that and make sure that you guys have time to ask one question. Here are all the people who have helped. They've been really generous, and again, I apologize for not being qualified to be up here, but I appreciate your time.